Great, okay. Let us start. We will not wait for people. So, hello everyone. My name is Vadim, and I am privileged to start the second section of our amazing meetup. The section focused more on the technical parts of things. So I have quite interdisciplinary background. I'm currently a researcher at Professor Fabian Theis' laboratory at Helmholtz Munich, where I work on single-cell immunomics and transcriptomics for predicting immunotherapy response, for patient stratifications, and all the different, these tasks around the immunotherapy development. In parallel, I'm a director of machine learning at American-French startup Essence. This is a startup focused on menstrual health, and I'm responsible for all things related to machine learning, large language models, infrastructure, and so on. So I'm the person who asks, is this solution scalable or not? And I do quite a lot of different ecosystem work. So, for example, Agata already shared that we organized a Tech Bio Meet-up in Warsaw just 2 weeks ago, actually. And I'm a co-founder of Data Nomads, a community of experts bringing together expertise from expert communities and local communities with the focus on machine learning. And today I will share you a glimpse into the work I do at Professor Fabian Theis' laboratory. So the first question is that, why are we doing this? And the problem is quite interesting, is that when you administer immunotherapy to patients, patients often get quite complex or even sophisticated response in their organisms, right? In their bodies, and that's why we need to look for biomarkers, some sort of factors that could predict what will happen in this or in that patient, and that's quite important, because some immunotherapies may work like ideally for some patients, but may be completely deadly for other patient groups. And I mean, we need to figure out, right, if we really want to help people. But again, the question is, why is so? Who is the culprit of these very complex responses? And our culprit here is immune system, and immune system is a quite complex, sophisticated network of immune cells which orchestrate the defense response to attackers, right? And again, this is a very complex system. It's quite hard to predict, like what will be going on in the organism, and that's why one strategy to explore it is to look at multiple biological layers at the immune system, and that's why we need multiomics. Multiomics allows you to look at the biology, at the organism, from different perspectives, from different views. You could look from proteins, you can look from the perspective of gene expression data, from receptors on the surface on immune cells. And today I will focus more on this part, on the single-cell transcriptomics related to gene expression data, and on this part, on surface proteins on immune receptors. Now, so I dove quite fast, but maybe you have some questions regarding, multiomics, or maybe immunotherapies or something like this. So if you have any questions, please feel free to raise your hand. I will stop and pass the mic to you, no problem. I will start with single-cell transcriptomics. You you're probably familiar with next generation sequencing. Or maybe, please raise your hand if you're familiar with next generation sequencing.
Heard the word.
You heard the word? No, please don't raise your hand. You heard the word. Okay, change the plans, right? What is the next generation sequencing? So in short is that, look, you all have cells, right? I hope you all have cells. What we can do, and there is something happening in these cells, like there are some genes, proteins, whatever. So what we can do is that we could lump all these cells together, like immune system cells, T cells, some specific T cells, whatever. We could lump them together. We could pre-process them, not computationally, but rather in a laboratory, to extract the necessary parts of the cell. So, for example, we could extract something that will help us show the light on how the genes are expressed in all these cells. So we pre-process all these cells, we lump them together. It's called a library. And we throw this library into the sequencing machine. And as the output, we get, I will skip some steps, right? But we will get the list of expressed genes and the level of expression of these genes in the input data. But here's the catch, is that, look, we didn't differentiate between those cells at all. We just have a sum of gene expression for all different cell subpopulations, cell phenotypes in your input library. So in this case, you will get quite fast. You can get the overall, not a landscape, right? But you know, this high-level picture of what's going on. Maybe you could catch some anomalies or something like this. But you lack the information about specific cells. And that's why we need single-cell transcriptomics. So with single-cell transcriptomics, we do some smart library preparation where we separate cells and we sequence them together, but we separate them before that. So as the output, we can get an information of expressed genes for each individual cells. Imagine that you get a date of 100, well, I mean 100 maybe too much, 50K cells, and you have the full gene expression data per each cell. So you could explore all the different complex phenotypes, subtypes of the cells and how they interplay and how they react to your immunotherapy that you administered before. And their data would look like this, is that you have your genes, right, gene 1, gene 2, and so on, and you have your cell identifiers. And in this matrix, in the cells of this matrix, you have the gene expression data per pair of gene and cell. At the end of this bioinformatics and machine learning process of analysis, the data, we get those wonderful pictures of the cell landscape, and each dot, each point here is a cell, and on the right side of the plot you can see the different cell clusters, cell types. We assigned them after the bioinformatics or machine learning work. So, and that's our target here for immunotherapy development, is that we identified those cell subpopulations, those cell phenotypes, and after that, we want to figure out what is happening when you administer your immunotherapy. What are the differences between patients with full response, with partial response, or sadly with no response at all. And I want to share with you a quite fun publication where we look for, biomarkers of immunotherapy response, and those biomarkers are genes that are significantly expressed in one cohort in contrast to another. So, to put it simply, we want to find genes which are significantly expressed in responders in contrast to all other groups. Here you can see the overall pipeline, right? So we get the sequencing data, we process it. Those are our beautiful plots with all the cells, and afterwards, what they did is that they applied the XGBoost. I think they applied here. They applied XGBoost to their data, and I want to highlight how they did it. So the data looks like, look, we have patients, right? And we have different cells for these patients, and we have gene expression data for each cell. So it's quite complex hierarchical data. But the authors here decided to go with the simplest approach, and it works. So what they did, they just simply trained the XGBoost model. This is a random forest machine learning model, ensemble of trees. And they, instead of predicting labels at the patient level, so instead of predicting responder, non-responder, they predicted those labels on the cell level. So, okay, this cell with this gene expression data, it came from the non-responder. Okay, great. XGBoost, please train on this or predict or whatever. And this cell with this expression data, it came from the non-responder, right? Again, please figure out what's going on here. And after that, they just, I think they just averaged the resulting probabilities of responder, non-responder to get the patient level label, right? So again, we have 100 cells. 90 of them are, I don't know, 0.80% of being a responder. We just average this. We get more than 0.5 probability that this is a responder. Yes, we put the label, we put the responder label on this patient. So it works. For me, it was surprising, right? But this is a very nice lesson, is that maybe we should really always try from the most simplest methods, because if it works, this, well, not great, but good, maybe we should do it. And they got around 0.84 outgrowth, and here on the right side you can see in the middle you can see the significantly expressed genes that help us to differentiate between different groups, and on the right side you can see the different the classification accuracy for different cell subtypes. So different cell subtypes helps you more or less to differentiate between responders and non-responders. Now, maybe I should comment on the AUC of 0.84. So this is, well, no, I'll put it that way. We are recording, right? So I'll put it another way. So if your AUC more than 0.90, maybe you overfitted on something. Maybe there are some problems with the data because it's too high of a level, right? So it's good. It's good. So for single cells, it's quite interesting, right? So we just saw that we are working on different different cells as units of our computations, right? We put them into the clusters of cell subpopulations, and then those clusters of cell populations, the patients are consisting of those clusters. So this is a nice paradigm that I want you to remember. The next part will be focused on immune receptor part. Now, so immunology is quite sophisticated, so I will not give you a primer on immunology, but what I want you to remember is that we have different types of immune cells, and in particular we have T cells and B cells. And what's important here is that there are surface proteins on the surface of these cells, T cell receptors or B cell receptors. And those proteins are what helps your organism detect different abnormalities, right? Anomalies in your organism to launch the attack on the agents. And what's extremely interesting about immune system is that those receptors on the surface, they are generated randomly. Imagine that in your organism you have a random number generator that spills out some sequences in the hope that those generated sequences will work for viruses, whatever, in the environment you are living in. Of course, it's much more complicated, right? Of course, there are rules for generating random sequences. They're not that random, right? But what's important here is that those sequences are there are a huge diversity of those sequences, and you can see the sequences on the left side of this table. Is that right? So maybe you can't see. Okay. The point is that there are many of those sequences, and we could gather them together, and for sequences with the same receptor, cells for the same receptor sequences, we could count how often we encounter those receptors. And judging by how often we encounter different receptors, different sequences, we could try to figure out what is going on in the immune system. Because if some receptor is prevalent, is abundant, overabundant, then it means that there is some sort of immune response is going on, and by analyzing this, we could link it to something. And again, we could link it to immunotherapy response. I would like to share with you a publication on application of the deep learning to immune receptors. Now, yeah, I don't think you clearly see here. So on the left side, you have the representation of your immune receptors in a tensor. It's not a matrix, it's higher level. In the tensor form, where we represent every amino acid letter in a receptor as a vector. Then we represent a sequence of those vectors as a vector of higher dimensionality, and then we represent the whole set called repertoire of immune receptor as a very, very large cube, right, of those values. And then we apply the deep learning model to this data in order to generate embedding, a vector representation of the whole repertoire. Are you familiar with the term vector representation? The idea on the high level, the idea here is that, look, we have some biological object, receptor, cell, you name it, doesn't matter. And we want to translate this object to a mathematical language. We want to put this object, cell, gene expression, receptor, in a space with vectors, right? We want to encode it as a sequence of numbers. With the idea of similar sequences having the close numbers, the close distance, right? It's like with dots on the plot, right? If you have dots on your plot, and in a two-dimensional plot, I mean, if you have dots on your plot close to each other, right? So the distance being quite small, it means that, okay, this is interesting, right? Those objects are quite similar to each other. Now imagine 300-dimensional space, right, where your dots are in this 300-dimensional space, and you try to figure out how to translate sequences to this dimensional space and how to compute distances between those points in this dimensional space. So again, this is another very important technique. I really want to highlight. If you don't know it, I really want you to remember it and look at it, because this is the foundational of all machine learning, deep learning work, right? Vector representations, embeddings. Just Google it and somehow understand. And we not only translate the receptors into these vectors, into this sequence of numbers. We translate the whole repertoires into a sequence of numbers. And deep learning models, neural networks, those are mechanisms for translating objects of one nature to this mathematical language. So yeah, it's not, deep learning models are not this fancy stuff or something like this. In reality, it's just a way to translate data of some nature to a vector representation. Because after that, we could work with common mathematical functions, tools, algorithms on these vectors. And what's quite interesting about this article is that they not only created those representations of different receptors, of repertoires, and were able to predict the immunotherapy response. They did some sort of a retrospective. They looked at the labels of the repertoires, of the sets of immune receptors with labels responded to immunotherapy or not responded to immunotherapy. And then they went back to the input sequences to figure out what are the key characteristics of receptors which affect the final label in the most significant way. And they found that there are different sequence motifs in the input data, and you can see they are highlighting in red or orange or something like this. Those are sequence motifs in the input immune receptors, which are well not linked but correlated, which are correlated with the prediction of different labels, responded to immunotherapy or non-responded to immunotherapy. And after that, in the ideal world, after that you start some very interesting biological work, experimental work, because after you know those sequence motifs, you could try to figure out how to filter out those receptors, how to explore them. You could apply those. You could filter out those receptors and understand their behavior in the laboratory setting, in vivo, in vitro, whatever works for you, right? Whatever needs to do it. And this could be a path to immunotherapy design, because using this model, you in fact you figured out what immune receptors are attacking, well, ideally, attacking the tumor. But okay, maybe we just need to take this receptor, and that receptor, the top 3 receptors. We could clone them and put back into an organism, into a patient, right? That's how you get to T-cell receptor therapy. One of the most prominent cell therapies as of today. It's a little bit more complicated than that, but you got it. So yeah, you see, it's like, again, right? It's not about just bioinformatics. It's not about just machine learning. If you have this interdisciplinary knowledge, if you work in interdisciplinary teams, you can do outstanding things. You can really work not just for the sake of new publications, right? But you can work for the sake of new publications and designing new medicines, which, maybe a little bit better, right? So we started a little bit later, so I will take a couple of minutes. So I hope you remember this graph from the single cell transcriptomics section. Now, right, so we on the level of scale, we go from small cells to the level of patients. For immune repertoires, for immune receptor, for immunomics, it's different. We start from the patients we look at immune repertoires. We usually don't look at the different immune receptors. We look at the immune repertoire as a whole, right? We're interested in computing statistics or applying models to the whole immune repertoire, because we want to understand the whole picture, right? The landscape. But with the model we just discussed, you can go back to the immune receptors, right? So the deep learning model we just discussed, it started on the patient level. It's just its input is just immune repertoires, just all sets of sequences. But thanks to some interesting retrospective analysis, they were able to go back to individual receptors with the hope of developing new immunotherapies. And I think that's quite important, because that's not how we usually do things in immunomics. The question is, can you do both? Yes, of course you can do. And the final publication I want to share with you is related to using both data. Again, the task is the same. Can we predict somehow? Can we find some markers of people responding versus not responding to immunotherapy? And they did quite nice, quite elegant analysis. They started with immune receptors, and they tried to find what immune receptors, what sequences are more frequent in responders in contrast to non-responders. So it's like, okay, this sequence, for some reason, we encounter it much more often in responders in contrast to non-responders. Okay, that's already interesting, right? But after that, they went to the single cell profile, to the transcriptomics profile of the same cells. Because in this case, our data is both gene expression data and immune receptor data. So we can go back to the gene expression data and understand what are the transcriptomic differences in those overexpanded cells. And they found gene signatures, some genes which are more expressed in responders in contrast to non-responders. And that's quite important, because if we understand all the pathways, if we could understand what helps immunotherapy, right? What pathways, what interactions help immunotherapies, and what doesn't help, right? What inhibit the immunotherapy, we could design more complex immunotherapies, right? We could design some additional techniques to help immunotherapy. I mean, we could design combination therapies, right? And that's why it's crucial to look for different biomarkers. And I mean, again, it looks like it's quite straightforward, right? It's not very straightforward, but I would say this is a great opportunity on the intersection of different domains, and I'm pretty sure you could find something in your work as well. And yeah, that's it. Thank you so much.
Okay, so you talked about vectorizing like different kinds of biodata, right? Like, and in depth you spoke on the receptor level, like I understood it correctly. Are there ways also to like vectorize like higher abstraction biodata, like you were speaking also on cell level, for example, like how does one vectorize data that's like working with whole cells seems kind of abstract for vectorization. So does one label stuff, or does one, do you understand what I'm asking, or?
Well, maybe we should just, I feel like this question is much more complex.
Okay, so maybe not.
Maybe we should discuss it afterwards, right? But the whole idea is that you need to, the methodology here is that, look, you need to figure out the very first, no, representation is not the right word, right? So look, for sequences, for example, right? What people do? You encode your letters, right, in some way. maybe one hot encoding, whatever, right? And then you represent your sequences as a vector of those vectors, right? That's how you move from sequences to, well, vectors, right? And there are, and it's like, saying, all right, garbage in, garbage out. And not only it applies to the data you generate in the laboratory, but it also applies to how you view your data and how you present it and what models do you use. The keywords to Google inductive bias, for example, right? Because different deep learning models, they have some assumptions on what the data look like. Let's put it that way, right? So you see, this is an interplay between data generation, nature of the generation data, and generated data, and the models you plan to apply, right? So that's why I said that this is a complex problem. Let's discuss it. Great, but I hope I provided some insight. Thank you.
So I also have a question.
Okay.
Because what I can see, even in Poland, is that we have more and more bioinformatics, data science, biotech startups interested in immunotherapies and in developing technologies for designing immunotherapies or supporting immunotherapies. And you also have very extensive experience in the startup realm. What are your thoughts on where this field is going, not only academically, but also commercially? What are the challenges? And do you think we're going to have some major breakthroughs thanks to bioinformatics and machine learning anytime soon that will push the pharma forward?
Right, that's a tricky question, right? That multi-layered question.
I only have those.
Yeah, I got it. Right. Again, I'll be happy to discuss it, to brainstorm it afterwards in more detail. But I would say that it's crucial to have success stories, because that's how you negotiate with stakeholders, with grant agencies, with regulating agencies, right? You need to show them success stories. So, I mean, if you would look at success stories, you will get the picture of what's going on, right? And what I personally do is that I on the high level, I want to highlight that this is on the high level, right? I look at success stories of the domain, tool, whatever, and the timeline passed after the insurgence, right? After the creation of this tool, or maybe something like this, right? And you can see that, okay, look, if there is one success story in 10 years, right? Is it good or not, right? I'm not sure. So yeah, so instead of answering your question directly, let me share my very simple methodology, how I do things. But I will have to discuss. Thank you for your question.
Sure, thanks a lot. Do we have any more questions somewhere? Okay, thanks then.
Thank you.